Hello and a very warm welcome to everyone joining us for today's Select Science webinar entitled Increased Confidence of Wound Healing Assays by Assessing Cell Migration and Cell Viability. My name is Dr. Lois Manton-O'Byrne and I will be moderating today's presentation. I am delighted to introduce today's speakers, Dr. Katrin Fletcher, who is an application scientist, and Dr. Michael Feitel, who is a market manager at TCAN. Katrin has a background in molecular biology and immunology and has been involved in the development of the Infinite as well as the Spark multimode reader series. Whilst Michael manages TCAN's multimode microplate reader portfolio, and they will both be explaining how the Spark multimode reader can be used to monitor cell migration in the context of wound healing, as well as discussing some of the key indicators of cell viability and metabolism. Just before I hand over to our speakers, I would like to mention a couple of things. Please feel free to ask any questions for the Q&A session at any time during the webinar by clicking on the Ask a Question button at the bottom of your screen. You can view the presentation in full screen by clicking on the tab at the bottom right of the media window player, and you can download related documents in the Resources tab at the bottom of your screen. The on-demand link will also be provided in a few days after the webinar. Without further delay, I would like to hand over to Katrin and Michael, and I would like to thank them for presenting to us today. Please go ahead, Katrin.
Thank you, Lois. Hello, everybody, and thank you for joining us for today's webinar. It will be about analyzing cell migration and wound healing with TCAN's new multimode reader. And I would like to present to you the results of our latest application project, which was a cooperation about cell migration and wound healing assays that was established on the SparkWE platform. To give you a little outline of what I will be talking about, the first goal of this project was to test the application of the SparkWE for a so-called gap closure or cell migration assay. And our second goal was to test the usability of confluence as an indicator for cell viability and cell growth. And we did this by correlating confluence to a luminescence-based ATP detection assay. And I will be talking about the assay setup, the measurement settings, and the results of our experiment. So just a few words about the scientific background of this study. Cell migration was in the center of this project, and it is a central process in the development and maintenance of multicellular organisms. For example, cell migration is important when looking at tissue formation during embryonic development, for organ formation and immune responses, but most importantly, in this context at least, for wound healing. So in the context of wound healing, cells of course need to migrate into damaged tissues, and they need to repopulate the area of the cut, of the scratch, or of the damaged tissues. Any abnormalities in cell migration can result in disease onset, including arthritis, atherosclerosis, and also cancer. When scientists want to study cell migration, they usually do this via manual cell imaging or visual cell imaging using common microscopes, either standard bright field microscopes or fluorescent microscopes. But this is a very time-consuming and laborious process. Imagine if you have to analyze dozens of different cell samples, then this is really quite a cumbersome process in the laboratory. So any type of automated imaging can help to increase the throughput of the experiments, and it also helps to standardize workflows. So what are researchers analyzing under the microscope? Mainly, they analyze confluence. Confluence is the determination of cell density on a certain growth surface. Normally this is the surface of a cell culture flask, a microplate well, or any type of growth surface that is used in the experiment. Importantly, confluence is not the same thing as cell number. Confluence, in contrast, is an indicator of the percentage of cell-covered area on a certain culture surface. The level of confluence drastically influences the growth rate, the metabolism, the gene expression, and many other parameters of cells. So it's highly important to start your experiments at similar confluence levels in order to get reproducible experiments and reliable results. The SparkWE platform is capable of measuring confluence in cell cultures. It has an imaging module that is based on an integrated bright field function that enables the real-time and label-free assessment of cell confluence in microplates. The Spark can also do cell counting in dedicated cell chips, and it can also assess the viability of cells using trip and glue staining. In terms of confluence assessment, the reader's integrated camera can detect any cell-covered area in 6 to 96 well plates, and a software algorithm will automatically determine the confluence ratio in each well. In terms of automating cell-based workflows, the Spark can also offer environmental control features such as temperature control, a dedicated humidity cassette that serves to maintain a humid atmosphere for cell samples, and it also offers gas control integrated right in the reader. All of these features enable long-term and fully automated live imaging assays. The Spark itself is not a dedicated imaging module, so it's not a big microscope, it's not a fluorescent imager, it's not a bright field imager. In contrast, it's a multimode reader. It combines many different functions and features under its hood. I have summarized some of these features here on this slide. Of course, it comes with all the standard measurement modes like fluorescence, absorbance, luminescence, and so on. It has some additional features, as I said, a gas control. It has a scanning function. It offers alpha technology, and so on and so on. Specifically, what we are interested in the context of this project is the Spark's cell imaging and confluence function. So this is just one small part of the multimode functionality of the Spark that was used in this project. In this cooperation, we teamed up with the Paracelsus Medical University here in Salzburg, and we raised 4 questions that we wanted to answer in this cooperation. First, can cell migration be measured reliably with the Spark imaging function? How can the environmental control features be optimally used for analyzing cell migration? Can a standard wound healing assay be implemented on the Spark 20M? And can confluence be normalized to cell viability? So these were the starting points. These were the goals for this cooperation. And I would like to give you an overview of what we found out here. We used the Radius Cell Migration Assay, which is a commercially available assay system based on a so-called gap closure principle. It is very convenient and user-friendly to measure any 2-dimensional cell migration. The assay is based on specifically designed microplates that have a spot made with biocompatible hydrogel right in the middle of each well to create a circular area which is cell-free, and this cell-free spot or the gel spot can be removed in the course of the assay to allow the migration of cells into this cell-free area. And this is what we tried to monitor in in these experiments. It permits the qualitative and quantitative real-time analysis of cell migration into the gap, into this artificial wound right in the middle of the well. We employed the following experimental setup for our experiments. We used two biliary tract cancer cell lines. We call them CL1 and CL2 in this experiment. The cells were seeded into the radius assay plates at an initial density of 30,000 cells per well, and the plate was then left in a standard incubator to give the cells some time to adhere to the surface. So we left them in the incubator overnight, and at the beginning of the experiment, the gel spots of the radius plates were removed so that the cell migration could begin. We tested a manual setup where the plate was kept in a conventional incubator in between the measurements, and the plate was only transferred into the Spark for the direct readout at an initial time point 0 after 2, 4, 6, 8, 24, and 30 hours respectively. And the second setup was the automated setup, where the entire measurement was performed inside the instrument, inside the Spark 20M, using all the cell features that the instrument offers: environmental control, temperature control, CO2 levels at 5%, and the humidity cassette for optimal humid atmosphere. The measurement settings were as follows. We performed a kinetic confluence measurement over the entire analysis period, so over 30 hours, with one measurement every 30 minutes. The measurement, or the imaging measurement, was done right in the center of each well, where the cell-free measurement spot was localized. So you see the parameters of the measurement here in this table. So in total, those were 60 cycles during the kinetic measurement with an interval of 30 minutes. So you would get one image every 30 minutes. With the central measurement spot, as I said, it measures right in the center of each well, and the confluence will be given for this fraction of the picture. The central measurement spot is ideal for quick checks of cell density, because if you take just one central picture, it will be the shortest possible measurement time. If you want to monitor the entire well, then it's recommended to image the entire well using the whole well imaging function. But for the purpose of this experiment here, where we knew that the area of interest was located right in the middle of the well, the central measurement pattern was sufficient. Importantly, the radius plates, or the microplates that come with the radius assay, are produced to have the measurement of the cell-free spot right in the middle of the well. In some cases, the cell-free area can be decentralized due to production variations. In these cases, it's recommended to do a whole well analysis or to use a user-defined measurement pattern to actually be able to image the entire area of interest. So you should make sure, or the researcher should make sure, that the imaging area corresponds to where the cell-free spot actually is. Importantly, there is no way to move the imaging spot after the measurement has been completed, so it has to be checked before the measurement whether the spot of interest, the cell-free area, is really located right in the middle of the well. We obtained the following results with cell line 1 and cell line 2. We knew beforehand that CL1 and CL2 would have a different migration potential. CL1 is known to show very little migration, while CL2 is described to have a high migratory potential. And we could confirm these assumptions. If you look at the results summarized here on this slide, you see that CL1 really shows negligible migration over the entire analysis period of 30 hours, while there's a drastic change when using CL2 and analyzing CL2 over 30 hours. The plots here show percent cell-free area. This is why we see decreasing curves with increasing migration. And here on the right-hand side, the red curve symbolizing CL2, you see continuous reduction of the cell-free area, so increasing gap closure, increasing wound healing in the period of 30 hours. We saw the same general trend with both the automated and the manual setup, which is shown here in these two pictures and in these two plots. You can see the progressive gap closure by CL2 very impressively when looking at the confluence pictures that are taken with the Spark software, starting from 100% cell-free area at the time point 0, progressing to around 44% cell-free area after 30 hours. You really see progressive gap closure, which can be nicely visualized by the Spark control software that comes with the instrument. We have also created a little video. These are just 8 pictures that serve to give an overview of the progression of gap closure. But since we took one picture every half an hour, we have created a little video with all the data points, which can be seen here. So what you can see here in the time lapse is continuous gap closure by CL2. Okay, some considerations for setting up the assay and for the handling of the assay system. As you saw in the pictures and in the tables that I showed on the previous slide, I showed a percent cell-free area instead of showing direct migration. So how did we determine the amount of gap closure based on the confluence value that is detected by the instrument? Well, the first thing to consider is that a cell number should be used that is high enough to produce total confluence in the area of interest. In our case, in the center of the well, which was the selected measurement area. So the suitable cell number should be predetermined to really get an entirely confluent well, with the exception of the cell-free area that is created by the gel spot. In our case, we used 30,000 cells per well, which was just fine in all wells and in all cases. Then the instrument will record the confluence in the area of interest, in the central image that we selected, and this represents either 0% gap closure in the starting situation or 100% cell-free area. In our case, we chose to set this to 100% cell-free area because this is what we wanted to monitor over time. That means over the analysis period of 30 hours, our signal of interest would decrease. In our case, from time point 0, 100% cell-free area to 75% cell-free area after 15 hours, and to approximately 44% cell-free area after 30 hours, which was the end of our experiment. It just depends on the desired results. So anybody wishing to recreate this experiment can either do it in the same way as we did, showing the cell-free area, but also the direct confluence given by the instrument can be shown, or the percent migration. In this case, this would be the inverse value of the cell-free area. So 100% cell-free area would represent 0% migration, and of course the other way around. Another thing that was important for us to address during this experiment was we wanted to discriminate between cell migration and cell proliferation. Of course, cells will divide over the analysis period of 30 hours. We just wanted to see cell migration, so we had to find a way to stop the cells from proliferating during our analysis period. This can be quite easily done by removing serum from the culture media during the analysis period. So we've removed the FCS from the media, which will not kill the cells, but it will preferentially lead to migrating, or will show us cell migration in contrast to proliferation. So the cell number itself will stay more or less the same, and the trends that can be seen where the cells will migrate into the cell-free area will really represent migration rather than cell proliferation. The second part of our study was aimed at showing a correlation, or the possibility of a correlation, between cell confluence and cell viability. Assessing cell viability is crucial for cell-based research because, of course, if you're doing long-term experiments with cell cultures, you have to make sure that your cells are viable and healthy throughout the analysis period. Normally, this is either done by employing fluorescence signals, for example, by GFP transfected cells. Sometimes the protein content is analyzed or the ATP levels are determined. In all of these cases, you need a second assay to assess the cell viability, either by protein content, or you have to perform a second measurement to record the GFP signal of the cells, or you have to perform a costly luminescence-based ATP assay to get an indicator for cell viability. So we raised the question whether cell confluence assessment can be used as an alternative for the determination of the intracellular ATP content. So we used the cell cytoglow luminescence cell viability assay by Promega to determine the cellular ATP content, and in parallel we recorded the confluence of our cell samples to have a direct comparison between these two signal types. And what we found was that there is a direct and very nice correlation between confluence and ATP signal for all the cell numbers that we have tested. On this slide here, I've summarized 4 example cell numbers that we have tested, where the blue lines represent the confluence values and the red lines represent the ATP levels that were detected using the cell cytoglow assay. And what you can see here is, in all cases, a very nice correlation between these two signal curves, confluence and the luminescence readout of the ATP level determination over the entire analysis time after seeding. So this was the result of the second experiment, showing us that since confluence and ATP signals exhibited very similar dynamics over the analysis period, we knew that since both signals directly correlate, that confluence recording can actually be used to replace a laborious and costly ATP assay that needs to be done in parallel. So this is a non-lytic and label-free way to replace ATP quantification assays with a very simple and real-time way to detect cell viability and cell growth. Importantly, just one consideration for the experimental handling. when cell numbers below 3,000 cells per well are used, the confluence signal is outside the recommended range, so below 10% cell covered area in the well. So this would be a cell number that is not recommended from our side because it's just too low for reliable results. So just make sure that the cells will have a value of higher than 3,000 cells per well for reliable results. That's really advisable in all cases to optimize the cell number beforehand, right before the experiment. So to summarize this again, based on our two experimental goals in this study, we saw that the Spark multimode readers are very well suited to assess cell migration and area repopulation using one selected gap closure assay, namely the Radius 96 well cell migration assay. Based on the Spark's integrated imaging module, we were able to perform reliable and real-time monitoring experiments about cell movement and the migration behavior of the cells. And furthermore, we were able to correlate the confluence value with the cell viability as indicated by the cellular ATP content, which really simplifies cell-based workflows and is another step towards automation and standardization of standard cell culture workflows and assays. All of the results that we have obtained with this study are summarized in our latest application note, which is called Monitoring Cell Migration and Wound Healing. It describes the implementation of the Radius assay on the SparkWE. It describes all the measurement parameters and the instrument setup and software scripts to obtain reliable results in both experimental cases, the wound healing assay and also the correlation assay between ATP levels and confluence signals. The application note is available at www.chicken.com/spark20m. And with this, I would like to hand over to Michael, who will tell us a bit more about the Spark readers and their integrated functions.
Okay, thank you, Katrin, for this nice presentation for the wound healing and cell migration. And what I'd like to do is basically go over the features and benefits of the Spark platform actually offers for our customers in terms of cell readers. But actually, it's even more than that. As you have seen previously during the presentation, I will focus on some of the key benefits associated with cell-based assays and what is behind the actual platform. So clearly, the cell module is available not only for the SparkWE, but also for the SparkWE. And the situation is the SparkWE is a mid-range reader basically for academic research and medium throughput applications. And we have developed now the SparkWE for more high-end detection up to 1536 plate format and with ultimate more sensitivity in terms of sensitivity as well as assay development and high throughput screening. If you look then actually at the features what we offer for cell-based assays, what Katrin showed you, in addition to that, of course, TF-FRET multicolor luminescence scanning and fluorescence polarization are featured more for the SparkWE with higher sensitivity. And this is something I think what is actually the key benefits in terms of differentiating the two readers for our customers and for the segmentation in terms of what kind of assays are you running, what is your throughput, and you can basically differentiate it by selecting those options which is your need for your laboratory. If you then focus on cell-based applications in terms of a workflow, you clearly start with cell counting and viability checking and the cell suspension. And this has been implemented by actually using a consumable. The cell chips allow you to basically read the cell concentration in 2 chambers with only 10 microliter solution, but also gives you the capability of checking the cell viability when you actually run it through a try-to blue paradigm. This is the start of a platform section in terms of how you set up a cell-based assay simply by having an exact concentration which are then dispensed in the microplate. And if you have then basically dispensed your cells in a microteter plate well, then you obviously want to keep your cells happy, and this is done by integrated gas control. You can simply by software sections regulate the oxygen and CO2 concentration. And this, of course, mimics the physiological solution and helps you to maintain the cells to a long-term assay. And this is particularly important if you want to run kinetic assessments like cell confluence. And this is, of course, also a situation in terms of using the spark control, because we can use so-called condition, meaning you can run actually a detection assay, like an absorbance, for instance, detection only when a certain confluence level has been reached. So really to increase your workflow area in terms of running an automation protocol. So the humidity cassette as an evaporation protection has been introduced to really allow to reduce the evaporation over a certain time period. And you see here proof plots in terms of running a plate with or without the humidity cassette. And this really helps you to avoid the side effect and basically the edge effect of a microteter plate well. And this allows you to really keep the assay running very strangely and ideal for long-term experiments. And of course, in terms of going on with a long-term assay, if you basically use a humidity cassette or you have a lid on your microteter plate, then of course you want to remove the lid to basically run a detection through a microplate well, and of course this is capable to be established with an integrated lid lifter in the Spark. And this is of course ideal, first of all, to have a contamination and evaporation protection, of course. In ideal with the humidity cassette, of course, you can also have basically an uncompromised CO2 exchange by venting it, and it keeps you basically also as a feature to use your own microteter plate well. So you can simply put a lid on a microteter plate well and stick the metal plate on the microteter plate lid, and the lid lifter then automatically removes and holds the microteter plate well. You can inject compounds and then put the plate basically back into an incubation position, really for long-term assays. The injector system in that sense helps you to also get more reliable results in terms of keeping compounds in solution. For instance, consider a solution where you use DMSO solution, and of course, if you're not steering, then you run into a problem that precipitation might occur. And of course, when you basically steer a solution, then it allows to keep the solution more readily and the compounds into solution, and that of course improves the concentration when you dispense compounds in a microteter plate well. If it comes then to detecting, for instance, a GFP, or as Katrin mentioned, a luminescence-based assay, when we start with a fluorescence GFP reporter system, for instance, of course, in that sense, the SparkWE features an enhanced fluorescence optics, which allows to not only detect basically a GFP in sense for a fluorescence optic, but it allows to use the system for an assay development for high sensitivity because of the implemented features like the built-in dichroic mirrors, of course, and the flexible bandwidth, which allows also to use unknown compounds by combining a scanning feature and then set the exact wavelength of a specific bandwidth for best optimal results. And this also includes a high-performance photon reading because we use a lens-based optical system for cell-based assays. If you can go on in terms of the luminescence assay, what Katrin mentioned with the ATB, if you go to more elaborate detection luminescence assays like red assays, so obviously this is always based on filter-based systems. And what we have come up with is an intuitive system in terms of using 38 long and short pass filters in the system, and the overlap of these filters allow you to more or less freely define a certain filter-based luminescence readout. So almost every bread assays or home-brewed assays are covered, and so it's a future-proof system in terms of running luminescence-based assays in the Spark readers. And one key issue we tackled then is when we're looking at luminescence assays, I think it's a common understanding that luminescence assays are basically prone to have a problem with at different temperatures and getting different results. This is, of course, something we tackled, and the Spark we are offering now a so-called cooling system. The T-cool system allows to basically keep the ambient temperature constant, and you're not running into a different result because the temperature inside the reader creeps up during a certain time. So if you look at the plot here, what's shown even at the standard specification at +4 degrees ambient, you see that the results basically will change over a time period of 10 minutes roughly. And only when you basically use a cooling system like the T-cool, this allows you to have really uniformity in the experiments. And this is capable to be run in the Spark platform with the T-cool to get reliable results, particularly for luminescence assays, as we know, but also in general, as we know that temperature influences chemical reactions in general. Roughly 1 degree changes about 10%, causes a 10% shift in the signal. So at the end, what we basically can propose is, when we look at Spark readers, the Spark 10M basically in an academic situation, the Spark 20M really in biotech and assay development and screening. You get reproducible data. It's a tailor-based platform in terms to save up your time because you can automate workflows for cell-based assays, and it allows you to basically get flexible solution, and the modularity and the upgradability of this reader platform boosts your assays and your throughput for cell-based assays and beyond. And with that, we would like to finish this webinar in terms of the cell migration and wound healing situations, and the application note what Katrin actually mentioned will basically help you to set up such an assay with your reader, and we certainly look forward to your questions. Thank you very much.
Thank you, Katrin and Michael, for a very informative and enjoyable presentation. Now let's move on to the final part of today's webinar, the question and answer session. If you've got any further questions, please feel free to submit them, and we'll hopefully get to them by the end. First question, how can I determine the cell number via the confluence number?
Okay, I think that's a question for me. Thank you very much for that very important question. Confluence is technically not the same thing as cell number. Different cell lines can have different individual size and will therefore reach total confluence on any given growth area earlier or later than other cells. For example, HUVEC cells have an approximate diameter of 12 to 15 micrometers, while HeLa cells, they're a bit bigger. They are closer to 20 micrometers. So that means a 100% confluent well of HUVEC cells will have almost twice the number of cells as a 100% confluent well of HeLa cells. So what can be done to determine the approximate cell number is to use your cell line of interest and create a standard curve that correlates the confluence and the cell number at the time of seeding. So the cell number at the time of seeding is the only exact number that the researcher knows. So you seed the cells into a microplate well, and when the cells have settled and adhered, usually this will happen overnight, you measure the confluence and note down how many cells give which percentage of confluence. Of course, it's recommended to do this with replicate wells to have a higher reliability of the results. And importantly, this process has to be done separately for each cell type that you want to use. So a standard curve has to be created for each individual cell type because of the above mentioned reason that cells can have different individual sizes. And then you can use the standard curve as a means of correlating all your subsequent assays with the same cell line and extrapolate the cell number from the confluence value.
That's great. Thank you, Katrin. We've got lots of fantastic questions coming in, so thank you very much for that. Next question. Can I also use a fluorescence label to be imaged?
Another very good question. At the moment, fluorescence imaging is not possible with the Spark. The Spark has a bright field imaging system that enables cell counting, viability, and confluence. The latter can also be done directly in the well. Since it's a multimode reader, however, you can measure fluorescence in parallel to the cell imaging. In fact, we have quite a nice application note on our website about how confluence was measured in parallel with cellular GFP signals and how these two signals can be correlated or were correlated in a human cancer cell model system. So I would like to refer you to this application note to get more information about how to measure fluorescence in parallel to confluence.
That's great, thank you. We've just got time for one more question. You mentioned cell counting in the Spark reader. Why is this an advantage in comparison to a manual cell counter?
Well, obviously, automated cell counting will give you a much better reproducibility of your results and also a much higher throughput. The slide adapter that is used for cell counting in the Spark has 4 independent spots for cell slides, and each slide spot has 2 independent chambers. So you can analyze 8 samples in one go, and that's absolutely label-free. If you just want to count your cells, there is no stain, no label, no dye needed to do this in the slide adapter. If you want to assess cell viability, there's a very simple trypan blue stain that is used that will discriminate between alive and dead cells. So the throughput is the biggest advantage of the automated counting system. Imagine how long 8 samples would take to count in a microscope, and I think everybody knows it's common practice to at least count 100 cells per sample. So the counting function of the Spark is accurate even at lower cell numbers. And as I said, you can assess the cell's viability in parallel using this very simple trypan blue counter staining. So using automated cell counting really frees up time in the lab and helps to standardize your cell-based workflows.
Fantastic. Well, that's all we've got time for today. Thank you very much, Katrin and Michael, for today's informative discussion and presentation. Thank you to everyone joining us online and for your interesting questions. I hope you found this a worthwhile session. If we didn't manage to answer your questions, please feel free to email me at lois@selectscience.net, so LOIS@selectscience.net, and I will follow up with your questions personally. If you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days. Goodbye, and thank you once again for joining us.